Good afternoon, everyone. So I'm going to talk to you about the differences between genetics and genomics and what do midwives need to know over the next decade between now and 2030 as genomics becomes more mainstream. So as I discussed, I am a midwife and I've recently started a role in a Genomic Medicine Service Alliance, which is a GMSA, and this meant that I had to reflect on my own training needs. Prior to this, I had worked in areas of governance and also in bereavement midwifery, so I had to think about my training needs, but also what educational requirements there are for midwives as conversations about genomics become more prevalent in healthcare. So the North Thames GMSA has been established in addition with 6 other GMSAs around the country to embed the genomic medicine into mainstream care. So the North Thames GMSA is made up of the North London region, Hertfordshire and Essex, and working with the universities and healthcare partners and trusts to ensure that there is equity of access to genomic testing, education, and literacy. And also on this slide is around the aims of each GMSA. So just as a refresher, this is a focus of how we aim to go from, we talk about chromosomes, and that is a package of DNA material, and that is found inside each cell nucleus. And genes, these are the instructions for making proteins. And DNA is made up of different sequences of nucleotides, which is as much about at the science side of things that I will go into, but just as a brief refresher. But the big focus is to remember that at the beginning of every pregnancy, there's that transition where you go from 2 sex cells from 2 different humans, and they form a zygote. And that holds a unique combination of DNA, unless it's a monozygotic multiple pregnancy. And so this unique combination of DNA will inform how essential proteins are coded and formed to support an ongoing development both in utero and after birth. So whilst everyone has a unique combination of DNA, we actually share 99.9% of the same base pairs with each other. So this means that all the variations between human beings, whether that is coding for your eye color or coding for how if your body can create proteins to allow your blood to clot, all of that equates to 3 million differences in our DNA. So when we talk about a singular gene, that is the study of genetics, whereas genomics is the study of all of our genes and how they interact with each other. And one of the reasons why I was really excited about the genomic midwifery role is thinking about how this word genomics, which is a very, very new word in terms of scientific developments, is combined with one of a really, really old word, which is midwifery. And bringing those two things together was one of the things that really excited me about my new role. So to code the human genome, that the project was thought about in the 1980s and then started as a global project in 1990, and the first draft of the full human genome, how all of our proteins and all of our genetic material works together, was available as a first draft in 2001. And then what's really amazing is that in a 6-year period between 2013 and 2019, the NHS supported and rolled out the 100,000 Genomes Project. So that meant that there were 100,000 individual human genomes that were mapped as part of this project. And that led to developments and breakthroughs in both cancer treatments and management of rare diseases. So As midwives, we've got a professional responsibility to remain evidence-based. And as genomics is an emerging field, it's going to be important that it becomes part of our everyday language and that we're aware of the implications that it has for the people that we care for. So during pregnancy, there may be people who are being treated for cancer. There are families who have history of genetic diseases and newborns with rare conditions. And all of these groups will have maternity input. So it's really important that midwives have a fundamental understanding of genomics. And it's also, it was also really interesting and pleasing to see that there are part of the standard of proficiency for midwives that genomics is featured within those proficiencies for midwives being at the point of registration. And so thinking about different ways of genetic testing, one of the hurdles that I've come across both in this role and in a previous role working in bereavement and around fetal postmortems is the term genetic testing. And it can sometimes be used as a catch-all, but genetic testing is that there are many different types of genetic testing. And this is something that's really important to be aware of, both as a midwifery educator and as a clinical midwife, and that there are different specific genetic tests depending on the type of abnormality or concern that you are wishing to screen for. And I've just put a few examples on the slide so that you can see that there are different ways of testing our genome, whether we're testing the whole chromosome to see if there's been a complete chromosome, either deletion or an additional, so a trisomy. There's also ways to look for changes in or missing parts of different chromosomes. And also the most recent development is gene sequencing and the advent of fetal genome and exome sequencing. So that means that you're able to find issues on individual genes, which may be missed when you were looking at either carrier typing or microarrays. So one of the first transformation projects that I'm working on in my role is ensuring that the pathway for bereaved parents needing genomic input occurs in a really seamless way. And so this will begin, as I've previously mentioned, with the conversations that I had with families around postmortems and genetic testings in terms of why their baby has died and supporting future pregnancy. So I've seen both at a regional level and at a national level that there are variations in how these referrals to genetic centres take place. And so the project that I'm working on is to reduce that variation and ensure that there's a straightforward pathway. And one of the reasons why this is so important is that there was a study published last year that showed with exome sequencing, so that's looking at the whole sequencing and the genome of a baby who died in utero, if that baby had no chromosome abnormalities and no clear obstetric cause, then previously we would have said that that was an unknown stillbirth. But a study last year showed that sequencing those genomes can find around 9% could be attributed to genetic disease variants. So if you look at the most recent Embrace report, where there were 2,400 stillbirths in 2019, and what percentage of those are or were attributed as an unknown cause, that meant that there could potentially be a cause of death for over 65 families per year within the UK. There are different studies that show the percentage of stillbirths as unknown can vary up to 60%. So that number could be a significant increase in trying to find out why some babies die in utero. So there's also areas for further discussion. It is now part of NHS screening and subtrusts for the NIPT and NIPD, which is the non-invasive prenatal testing and non-invasive prenatal diagnosis. And we're also looking at how we can test the fetal DNA of it. of a woman who is rhesus negative to check if the baby is rhesus negative or rhesus positive, to be able to provide targeted rhesus, to provide targeted anti-D treatment. There's also areas for development around the testing for sickle cell disease and thalassemia, targeted cancer treatment in pregnancies, pre-implantation genetic testing, and also for a more nuanced conversations around consanguinity amongst couples. I would advise, if anybody has more interest in genetics and genomics within midwifery, then please get in contact. But there's also a really good genomics education program on the HEE website. And that goes through different bite-sized chunks of information that can be done at your own pace, whether you want to know more information about how cells divide and reproduce, or if you want to know about how to take a family history or about pharmacogenomics. There's lots of bite-sized learning on there, but I'm more than happy to talk to anybody about this topic in more detail. So thank you. I'm happy to stay on for any questions.